pagi semuanya, perkenalkan kami dari kelompok 2. Di sini dari kelas IKM 3C. Di sini kami akan mempresentasikan hasil naskah mini literatur review kami dengan judul atau topik The Body's Reaction of Nicotine from Absorption to Addiction. Selamat menyaksikan.
Before we dive into the specific on these presentations, let's take a look at the abstract. Nicotine is a psychoactive compound in tobacco products, produces complex biological effects from absorption to addiction. Recent studies highlight its pharmacokinetics, receptor interaction, neural circuits, and genetic or sex-related factors, with different products showing varied absorption and addiction potential. Nicotine influences multiple pathways, not only dopaminergic but also glutamatergic, GABAergic, and neural circuits, and affects processes like angiogenesis and immune function. Key research gaps remain, particularly for new products, understudied receptor subtypes, and long-term effects. Overall, evidence underscores nicotine's multifaceted actions and the need for further research.
Determining keyword. The process of determining keyword was carried out by identifying the core concept within each research question in the SPS, then expanding this problem using synonym, scientific terminology, and Boolean operator and/or. For LT-list discussion, the new biological methods of nicotine keywords such as nicotine addiction, reward pathways, and ACHRS and eukaryote were selected. Studies focusing on receptor subtypes used keywords like A4, B2, A7, and genetic variability. While biological and pathophysiological studies evaluate keywords like angiogenesis, immune modulation, and ACE2.
Saya akan menjelaskan tentang literatur search strategy. The review studies employed a systematic literature search using multiple academic databases like PubMed, Scopus, Google Scholar, and the others, with specific keywords linked to the research topic. They applied clear inclusion, exclusion criteria, focus on recent publication and prioritas, full text availability. Screening removed duplicates, and irrelevant papers. Following Prisma with lines, the process ensures transparency and reproducibility. The studies include both animal and human data, with meta-analysis or qualitative reviews conducted when possible, highlighting heterogeneity and research gaps.
Next, here are the type of literature. First, the review literature covers multiple study types, such as experimental studies, narrative reviews, and plot analysis, and so on. Second, the research uses multi-level experimental approaches, such as vascular relaxation assays, endothelial cell cultures, and cytotoxicity and biochemical analysis. Third, the analysis highlights key mechanistic findings, including the roles of specific nicotinic receptor subtypes, distinct neural circuits, pharmacodynamic differences, and variability in pharmacokinetic data.
Followed by a discussion of problem statement. Despite extensive scientific investigation, major gaps remain in understanding nicotine, its complex biological and behavioral effect. Exciting studies show inconsistent results due to different in-device design, formulation chemistry, user-preferring behavior, and research methodology. These inconsistencies also extend to nicotine as broader biological effect, such as its influence on immune regulation, cellular proliferation, and disease pathways, which remain fragmented or contradictory. Therefore, a comprehensive synthesis of current research is urgently needed to clarify nicotine's mechanisms of action and resolve discrepancies across the literature.
Okay, I would like to explain the concept and information obtained from the literature about the body's reaction to nicotine. First, we all know that pharmacokinetics refers to how nicotine is absorbed, distributed, metabolized, and eliminated by the body. The key concepts regarding nicotine include its molecular mechanism, involvement of neural circuits, kinetic and sex-dependent factor, and of course, from broader biological effects. Nicotine absorption varies depending on the product used, whether conventional cigarette or electrical cigarette with different formulation, which affect how much nicotine enters the bloodstream and how addictive it is. At the molecular level, nicotine interacts with specific receptor in the brain, which regulate its rewarding and aversive effects through complex neural pathways. Genetic variation can influence an individual's susceptibility to addiction, while differences between men and women influence receptor expression and behavioral response. Nicotine not only for giving an addiction effect, but also impact a cell proliferation, immune regulation, and disease processes. With understanding this interconnected aspect, help us develop target strategies to address nicotine addiction effectively.
For the background, nicotine can be absorbed through many types of products, and each product delivers nicotine at a different speed and intensity. This difference affects how quickly nicotine reaches the brain and how strongly it activates reward, withdrawal, and addiction pathways. Recently, the use of smokeless oral and synthetic nicotine products has increased, especially because they are marketed as cleaner or safer alternatives. However, they still carry potential risks, particularly related to cardiovascular and oral health. Another concern is that the long-term effects of synthetic nicotine remain unclear due to limited research. For the methodology, the studies we reviewed used a combination of human pharmacokinetic trials, molecular laboratory experiments, and narrative review. The human trial focused on measuring key indicators like Cmax, Tmax, and AUC to compare how different nicotine formulations are absorbed. Meanwhile, molecular studies investigated receptor activity, desensitization, and neuroadaptation to understand how nicotine affects the brain at the cellular level. The narrative review supported this finding by summarizing toxicology data, cardiovascular biomarkers, and regulatory information. Together, these methods help build a comprehensive picture of nicotine's absorption and its impacts on the body.
Pada slide ini, saya menjelaskan bagaimana empat jurnal menyajikan dan menginterpretasikan data terkait respon tubuh terhadap nikotin. Dari jurnal yang pertama, menampilkan grafik konsentrasi nikotin terhadap waktu dan membandingkan nikotin garam serta bentuk bebas melalui Cmax, AUC, dan Tmax. Dari jurnal yang kedua, menyajikan diagram jalur molekuler untuk menunjukkan bagaimana nikotin mengaktifkan reseptor NHCR dan mempengaruhi neurotransmitter. Dua studi lainnya menggunakan heat map, grafik ekspresi protein, dan gambar struktur reseptor untuk menggambarkan desensitisasi dan perubahan neurobiologis selama paparan nikotin. Secara keseluruhan, keempat studi ini mengumpulkan data visual dengan mekanisme biologis seperti proses penyerapan, aktivasi reseptor, dan pembentukan adiksi.
There are still mixed findings regarding nicotine absorption because the results depend heavily on product type, device design, and user behavior. For example, some studies suggest that nicotine salts are absorbed more quickly, while others report similar delivery between salts and free base form. In terms of biological pathways, the dopamine pathway is consistently linked to nicotine's addictive effect, but other pathways such as glutamatergic, GABAergic, and habenular pathways show inconsistent results across studies. Additionally, the long-term health risks of smokeless oral and synthetic nicotine products are still not well established, mainly due to limited long-term data.